back in a minute. You guys are probably writing too much, by the way. Alright, 3 more minutes. Alright, you guys ready to wrap it up? One more. Okay. Alright, we good? Cool. You guys know. Would you guys like to pass them to your partner? or not, I guess you don't have a partner, but why don't you pass them to your neighbor? Maybe might be a better word. We can sort of self-correct. Does that sound good? Okay, so thanks again for getting that stuff in, most of you guys. Sorry for any confusion with the upload system, if there was anything without getting access. I'm learning or trying these sort of new system out too, seeing how it works, and I'm going to do my best to make sure it gets easier in the future. Try to sort out some new approaches if we need to. But did it work okay ultimately once people got access to the drive files?
Still don't have access. I did email you.
Yes, you emailed me after the deadline of the quiz, which was midnight, and I was sleeping. So I will give you access this morning, but the deadline for submitting that stuff was midnight, which you might not have caught because we talked about that before you got here on the first day. So.
Yeah, because I sent an email out to everybody.
Perhaps I did not get it, or my email might have changed it, but we'll work it out. We'll work it out one way or another. These are really easy sort of little things that we can resolve because we're in charge and we can make it how we need it to be. So, sorry for any confusion along those ways, but we will sort it out and we will make it better. What do you guys think about making the questions?
Was that right?
Reasonable?
Yeah.
Was it hard? No. What do you guys think about your colleagues' questions as you took the test today? Good questions, bad questions, middle questions?
I thought they were pretty good, to be honest.
Okay.
I was worried about like what type of details people would have focused on, so on and so forth.
Which you knew, excuse me, you should have known beforehand because you guys had access to it.
Right. But I didn't. So I wasn't too sure like what kind of details, because I know everyone tends to focus on one thing or another depending on what their background is and so on and so forth. So I was like really like wondering what that would be like, and then everything seemed to be, well, obviously everything covered more or less.
It's pretty straightforward,
detailed, so
right?
Yeah.
Okay, cool. So yeah, just a couple things just to mention as we sort of move forward. Did you guys see any differences between, so maybe how you guys thought about the things that you thought about that were important to ask and think about versus some of the things that I thought were important to ask and focus on? What did you notice? I'm sorry?
Yeah, more conceptual.
Yeah, versus?
Definitions.
Yeah, so definitions are important to know. I'm not saying you don't know them, but you guys, I mean, part of the reason for the exercise, if you remember when we introduced the idea, was to let you guys basically start getting some practice because you guys will be instructors or teachers or teaching assistants at some point. And so I try to focus on the conceptual things more broadly because in order to get it right, you have to know the details that a lot of your questions were asking, right? So it's a way to basically double dip on a question and see, do they know the specific information as well as the conceptual? Versus if you just ask the specific, you don't know if people actually know the conceptual or not. So that's sort of one of the approaches that I try to take. So anyways, okay, so let's see here, we'll throw this on. We can walk through it real quick if you want. Hopefully it'll zip through. One of the things I tried to mention, if you guys remember a few minutes ago, is that maybe you're writing a little too much. So I just wanted to remind you guys, I know we never want to get anything wrong, right? So we always want to pack in everything that we know and all that kind of good stuff. But the idea that I want to shoot for through the arc of the semester is to sort of keep these sorts of exercises short and sweet and use them as prompts for discussion and things like that. But it shouldn't be too stressful or you feel like you need to pack in too much, because the idea is that a lot of the learning process hopefully should come from actually writing the questions rather than taking them and preparing for what your colleagues wrote as well. So the first question basically, localization of function versus distributed processing, right? So a lot of verbiage in here that you can just get rid of, right? Doesn't matter that there's a person sitting next to you on a plane or a bus or a train or whatever. It's there to sort of set the framework of the question, but don't let it distract you. basically it just comes down to localization, function versus structure. The reason I like these, or I use like a question like this, something like this, is basically it happens in real life all the time. Once you sit on a plane going somewhere, somebody starts talking to you while you're takeoff, and I go, what do you do? Oh, I'm getting my master's in psychology. Oh, cool. Let me tell you about all the problems in my life, and maybe you can help me out for free while we take this flight. And people are interested in the brain and the mind and all this kind of stuff, and they're really engaging, fun things to talk about because hopefully, right, we address timeless questions and things like that. The mind-body problem, things that a lot of other interesting, fun things to talk about, and people will engage us and ask. And we need to have sort of sometimes a short elevator answer as opposed to long essays, right? And so I just wanted to kind of use it as a way to do that. Obviously, sort of what I was getting towards is the idea that at least where we understand the field of view right now is that kind of represents a false dichotomy. It's not one or the other, of course. Hopefully we can see that it's a little bit of both. They work in concert together, distributed neural networks that tend to be evidently served by individual localized hubs of areas or smaller networks that are working together with broader distributed things. Hopefully the persons who you're marking discuss a little bit of the evidence for or against, because the person next to you on the bus ride here from LA would probably be more persuaded if you actually gave them a little bit of evidence. What would some of that evidence be? Okay, so broke is what would you say?
That supports some idea that the localization, that damage to that certain area can cause damage to language, but that's still not completely inhibits language, so that's why it's not totally localized. There's still general.
Yeah, and the other thing that's important to note too, right? It's not that it just damages language, but that it seemed to leave most other things intact, right? And then of course that seemed like, oh yeah, totally, let's dive down the rabbit hole of localization of function, right? But then how we kind of back it off and things like that. So any questions on that? Okay. Let's see here. Number 2. So what are some ideas, theories, things that were famously wrong about the brain and the nervous system, either historically or modernly, and briefly? Why were they wrong, and why were they important enough to be worth knowing about, right? And so here I've listed a whole bunch of things, because there was a whole bunch of things that we talked about, including the idea that the heart is the seat of the soul, the pineal gland is the seat of the soul. There's lots of places people have thought of the seat of the soul. We talked about the brain operating as a radiator, thanks to our philosophical friends of the Greek era, and talked about nerves operating as tubes and mechanical hydraulics. Ventricles being these small little bubbles in the brain that she kind of drew at first sort of inaccurately and then a little bit more accurately. Of course, we've talked about chronology, aggregate field theory, equipotentiality, neuron doctrine, which surprisingly I listed as not being true because we found that there are exceptions to it, right? Same thing, we have particular theory, localization views of brain function, holistic views of brain function, so the same answer to the question beforehand. Glial cells were merely glue that held brain cells together, or neurons together. Glial cells are not involved in neural signaling. We can make a much, much longer list. Probably a really cool book, actually, if you guys ever want to get together and write a book, make a little bit of coin, or make it an ebook. It's sell it for like $1. It'd probably make more money than the publishers would give you if you wrote a hardcover book. But there's a lot of things, right? And we hope that that list gets longer, because we hope that you guys are going to go off and discover something. And if you do, it means that you probably added to the body of knowledge that might correct, improve, or enhance what we might have known beforehand. So, any questions on that? Okay. What are some aspects of why is it important enough to know, basically, why do we waste our time talking about historical things that are not necessarily accurate? Yes.
I wrote that I think it was important because it challenges the zeitgeist. It also represents the zeitgeist of the era, and gives the opportunity to further research and further the knowledge of what was once considered dogma or a fact, basically.
Totally. Yeah, I mean, past is prologue, right? If we want to know where we're going, we should probably have an idea of where we've been. So that's kind of, anything along those lines suffices as answers. Obviously we don't want to repeat the mistakes of the past or waste your time trying to figure out if things operate as a reticular net, right? Because you can save a lot of thesis work. Hey, we already started that out, right? Save your friends some work if they got this really brilliant idea of testing that out. So work on that. And of course, we advance new hypotheses, which even though they are wrong, stimulate a critical discussion, like you said, represent some of the zeitgeist and brought about some novel ideas. So I said we've discussed 13 Nobel Prize winners in neuroscience so far, briefly identified 2, and briefly described the discovery insight associated with each of them. Might have been surprised that we covered 13, yeah, but we did, surprisingly. Not in great depth for all of them. They're all equal, some more equal than others in depth. And so any of them work. We talked about Watson and Crick with DNA, we talked about Sherrington in the synapse, we talked about Golgi, we talked about Hall, then we had a slide where we mentioned a few others. Not in great depth at all, but anything works. The motions we'll talk about, it's a husband and wife team, so there's 2. They actually worked on some things with spatial navigation memory. We'll talk about place cells later on in the semester. Sperry was into neurodevelopment with axon projections for the visual system. Eagle and Weasel worked on the vision system. Axel and Buck worked on open olfaction. And O'Keefe actually shared the prize together with them just for memory. So I think all of those were mentioned in class, but any of it works. It's kind of cool to know who the people are, just in case you happen to bump into them and, embarrass yourself, which has happened to me. I once asked somebody on this list on a bus ride that I was sitting next to, who I did not realize, and they were explaining their research, and we were sort of doing the traditional sort of bus ride exchange of, oh, what do you do? Oh, well, what do you do? Sort of thing. And it turned out we both studied hippocampus and memory, and I didn't recognize the person's face, and they were saying that they were having that it was getting more difficult to publish in their particular little micro niche of the field because it's become very popular. Without obviously alluding to the fact that it's popular because they just won the Nobel Prize, which, because it was a dark bus at night after a long day, I didn't put together, and I recommended that perhaps they might find more success if they joined the lab of the person who was themselves. Yeah, it was a lot of fun. We had a really good laugh over it.
Who was it?
I'll tell you later. Actually, no, it was one of the posers. It was, and yes, I got a really cool selfie from it. Because then he laughed. He's like, "No, this is great. I really enjoy the scientific conversation. Most people are just brown nosing and wanting to just get a selfie with me." And I'm like, "Oh well, now you realize I kind of have to get a selfie." And he obliged. We had some fun. And anyways, it's important to know the people, and obviously I knew the person. I just didn't put a name to a face. We'll talk about that deficit of memory later on in the semester as well, because we've all experienced that, right? Mm-hmm. Okay. So, those Nobel laureates. Yeah, you never know when you might bump into one. So, let's see here. So, some of the other questions you guys had. Cells that create myelin sheaths around the axons were oligodendrocytes and Schwann cells. And obviously, the purpose of myelin is to insulate the axons. And then, of course, the difference between Golgi's reticular theory and Cahal's neuron doctrine. Which one is correct? I added that. Okay, somebody wrote this, I forget who, this is somebody wrote it, but they didn't ask this, and I thought that was a really good addition to add, so I added it. And we talked about that a little bit already in here, so a little bit of both. Obviously the neuron doctrine is the predominant thing, right, but it's important for us to know the exceptions too, because it's really dangerous for us, particularly as scientists, to be caught in the trap of speaking in absolutisms. Right, because very, very few things in science are laws, right? Like Newton's law, thermodynamic laws, physics, things of that nature are incredibly rare. Mostly everything that we're working with are theories and models that have accrued a ton of evidence that are really good support, but there are usually exceptions to the rules, and so absolutisms tend to get us into a little bit of hot water. So anyways, and then of course I think last one is central dogma of molecular biology, DNA to RNA to protein, the translation and transcription. And then if you guys want, I'll post the rest of these. Well, you guys actually already have everybody else's questions and answers. I'm sure you might have taken a look at. So we don't need to go over them so much. And you guys have that available. Do you guys want me to post this as well? Or, yeah? Okay, I'll just post it up there for fun. Because I know that's will constitute fun weekend studying and things like that for you guys. So, if they answer the question and it was in the realm of what we were discussing, give credit for that. If they didn't, note that it was off target and give it back to them, and they can correct that and fix it, and they'll get credit.
So you do 6 out of 6? Is that what you're saying?
If they, I mean, well, I don't know. You tell me if it was 6 out of 6.
That's alright.
That's your job to grade it, right?
No.
Yeah.
That's one thing.
So remember, the broader idea for this too is to make sure it's to get you guys points, not to take points from you guys. So it's here to help you guys. So you know, my expectation is that everybody walks out of this with 100 today. And if that's not true, then talk to me afterwards, because you have the opportunity to fill in the blanks or to correct anything that was astray. So you guys can hand those in later, leave them on the desk after class. And so that'll kind of wrap us up for there. Are there any questions? Yes, ma'am.
For the exams, for short answer, you could have or just like 2 sentences, unless.
No, yeah, that's how I was like, oh,
Well,
it was, and that's what I
Unless you otherwise say a little bit more detail.
So it's a good question, and the most honest answer I can give you is that it depends, right? So one clue that I try to give people is how much space I give you to answer. So you notice that I've like shrunk them in, and I was kind of like saying, you guys are writing too much, because it's like if you have to, right, like squeeze a bunch of stuff in, then it might not be the same match with the space that was being sought after. So yeah, I mean, basically it'll be a little bit of both. So there might be some questions where you can answer in a couple words.
Not even like full sentences.
Well, it depends.
Right, it depends.
I mean,
But
I hate to say this, like, right, we already know these answers, right? And then there's going to be other answers for questions where, particularly if you're given space or, you know, the nature of the question, you know, you want to best represent yourself, which you guys already know as well. So there was, just as an anecdote, I remember studying in grad school for a test with my friends, and they were trying to just drill some aspect of developmental neurobiology in a thick skull, and it wasn't. We were working on it hard because the skull was pretty thick, and so they asked a question about how some receptors worked on cell membrane, and thought about it. It was a very, you know, we were practicing for an oral exam that we had to take. There was like 5 people in our class, and the exams were with the 2 instructors for the class, just you and a piece of chalk for an hour. And they asked you questions, and it was very challenging. And so, you know, we were practicing, and they gave me a question. I thought about it, and I had like little, you know, whiteboard marker in my hand, and I just said, oh, synergy. And I answered the whole thing in 1 word. So I mean, there's ways that you can be really, if it represents it right, then yeah. But also, no, these are quiz questions, not test questions.
right.
So, but hopefully the idea is that I'm not looking to trick you with anything, just very straightforward. So are there any more questions before we jump forward? Dive into some neuroscience? Awesome. Okay. So we were talking about neuroanatomy, and we finished off introducing some of these ideas here that we were looking at some. Oh, we should switch the orientation of the lights, shouldn't we? Where are those?
Sorry. I have a desk in front of me.
That one is probably less so. Is that better?
Yeah.
Okay. This way I can see you guys really well and see when I need to wake you up, and then this, but a little darker here. Okay, so we introduced this idea. We were looking at cross sections of the spinal cord in this regard. We talked about dorsal areas. We talked about ventral areas. We talked about the different roots. Basically, that phrase that was referring to the basically these connections of the nerves with the spinal cord. We talked about the white matter. We talked about the gray matter. We talked about, we'll introduce the idea that these are basically going to be referred to as horns. So you have dorsal horn, ventral horn, the dorsal root, the ventral root. Now, you can't tell from the, well, you can actually tell by the anatomy of the gray and white matter respectively, but at first pass, you might not know if this was the dorsal side or the ventral side. It could be kind of rotated. Like I said, that you would know if you knew actually a little bit more. If you, just looking at this will tell you it's dorsal or ventral. But basically the idea, just reminding you that this orientation is looking this way. This dorsal back here, so this is the dorsal side, we're looking in that direction. We introduced the idea that there are sensory nerves and motor nerves, so basically have information coming in and coming out. And we actually finished with questions about what was afference and efference. Okay, and there's a little bit of discussion about that. The way that I like to remember that is, so afferent is essentially what I mean by that. They're very, very similar. Afferent and efferent. Two very similar sounding and looking words. They're very confusing. They basically mean no different things. So you have an afferent nerve and an efferent nerve, and what it refers to are the nerves that are basically sensory and motor, or coming in versus going out. Now, in and out in reference to what? Well, in reference to the central nervous system, basically. So basically you have afferents that are basically your sensory neurons. Okay, that means the information is coming in, or it's going to the central nervous system, as opposed to the efferents that are coming out from the central nervous system and are otherwise conceptualized as motor neurons or something like that, for example. So lots of people remember them in different ways. I remember them by essentially, so afferent are sensory neurons, right? So sensory neurons are going to be the things that, like, when you sense a touch, for example, when you feel, you know, I don't know, coarse versus smooth thing, something, you know, you can feel that in your sensory receptors, touch perception. And so I think of that in context. So anyways, what makes me remember it is if you are affluent, right, rich, wealthy, you might be able to feel really nice fabrics, all the nice velvet and whatever, I don't know, cashmere or whatever fabrics they wear. I'm not rich, so I don't know if they go around feeling all their nice fur coats and whatnot, right? But afferent, sensory, affluent, very nice sensory reception. Is just a trick I use, but my brain doesn't have to work the same way that your brain does. You come up with your own memory joggers and things like that. But anyways, we talked about those ideas and the idea that we have these dorsal roots carrying the sensory information, the ventral roots carrying the motor information from the spinal cord, as opposed to the information into the spinal cord from the dorsal side. Okay? We even introduced the idea of a reflex arc that we'll talk about in a little bit more detail as we move in, which is that you have sensory information coming in, such as, ouch, I stepped on a tack. Right? So that's coming in from your sensory nerve on your foot, coming up through your leg. It's going to come in through the dorsal root. It's going to make a synapse on the motor neuron in this gray matter, because it's gray, which means it's not myelinated, so you have cell bodies there as opposed to axons. And that is going to send a projection back to the motor side to output your reaction, which is, oh, you stepped on a tack, pick your foot up, right? Now, what was nice about that was it provided an illustration of times when the nervous system there is working without a lot of top-down control. It's a reflex loop that doesn't have to go to the brain, right? So the brain's not involved in everything. And we know that we can, to fast forward to my favorite topic of memory, we know that the brain doesn't have to be involved in memory in all ways in that regard, because we can see times when the reflex arcs are adjusted outside of the brain in that regard. Does that make sense? Okay. So we talked about introducing those ideas. We can characterize the central nervous system, at least in the spinal cord, in different ways. I'm not going to talk a lot about this stuff through the course arc of our course, but it's just useful to introduce anatomically so that we do have an idea of that. So basically, we can look at different segments. We have the cervical, thoracic, lumbar, sacral. We can see the colors match up to the different sort of segments you can see corresponding to in the spinal cord. And so what's also interesting is that when you look at neuroanatomical slices of this, we can see that there's different morphologies of the respective balance between white and gray matter as you move up and down the spinal cord. So for example, this looks a whole lot different than this, doesn't it? If you look at the cross section, the black and white version, not the color part, but the black and white version, we can see that there's a different distribution of motor neurons, or the bodies that is, versus the axons in white. Whereas here, it's mostly all white axons. And so it basically represents these columns of neurons rising and descending through the spinal cord, and their respective sort of shapes as they move up. And we'll get into that a little bit later when we talk about motor and sensory systems. But I wanted to introduce that concept first because we will come back and revisit it. So, let's see. This is just another way to conceptualize and sort of dial into this idea that we introduced a minute ago. So here we actually have our spinal cord schematic reversed from what we looked at before. So this used to be that we were looking at it this way with the dorsal side back here, the ventral side here. Now you're looking at it this way. So the dorsal side is back here. Okay, now you have ventral roots. So dorsal roots, ventral roots. Okay, you have the dorsal root ganglion. So you have these sensory neurons. Notice that the cell bodies are here. Remember we called them ganglions in the periphery? Okay, now they send out their unipolar cells. All right, so they send out one projection, and you have receptors that come here that detect our sensory perception, and then projections that send that information here. And when they come in the spinal cord, they can do a couple things, right? They can decussate or split. Right? We talked about axon collaterals last class. We talked about how the different terminal boutons can sort of spread out. And so those collaterals definitely happen. And you can have sometimes when they rise up and ascend into the brain through basically these spinal columns that rise. They can also make these synaptic connections internally within that segment, just like we just talked about. And this is a representation of that, where they're going to project onto an interneuron. Remember, you asked about times why wouldn't you have myelination or examples of those situations. Well, here an interneuron is not myelinated. We know that because it's in the gray matter, not the white matter. It's going to project through that interneuron, which modulates this motor neuron, in which the body is here, but the projection comes out. Make sense? Okay. So we can see different ways we can categorize that. Okay, and if you look at it more broadly, just to give a broader picture, we have our vertebra, the bone here, of course, and we can see that this example that we were looking at here and on the prior slide is actually wrapped up in other sheathings. So we can see these sheets built upon it, and these ganglia, these roots, are actually over and over again up and down the spinal cord, as opposed to this singular example. So just a little context and scope there. Alrighty, so we talked about the somatic and autonomic nervous systems and those respective delineations a little bit. We'll dial into that a little bit more. We'll talk about the sympathetic nervous system. So remember, when we talk about the sympathetic nervous system, okay, we're talking about the branches of the autonomic system. So we have this sort of tree here. We're in the periphery, so that's all this yellow area. We're talking about the autonomic side, and we're going to break that down into sympathetic and parasympathetic. Okay? And so when we talk about the sympathetic, okay, we're basically talking about that fight or flight reflex. Okay? So this is sort of a helpful little schematic to show that respective delineations between the sympathetic nervous system on this side and the parasympathetic. Okay? So, for example, we could just talk about, say, pupil dilation. So the sympathetic nervous system is going to be involved in dilating your pupil, whereas the parasympathetic is involved in basically constricting your pupil. Different things in different times. Basically, they work in complementary systems. One's basically an activation system, and the other one's sort of a relaxation system. Okay? So fight or flight. let's see what I'm looking for here. Where is the yes, okay. Fight or flight, sympathetic nervous system. What does it do to some of the digestive tracts? For example, if you're in a situation where you're in a fight or flight situation, would that tend to be a situation that is scary? Maybe, it might be, which is why you might think that some people pee their pants, because it relaxes the bladder. It is sort of outside of our otherwise conscious control. It can be very quick. Now, of course, we can control those things, I hope, for the most part, right? But there are times when it happens, right? That's why. It's under control of that sort of sympathetic nervous system, fight or flight. And of course, when it relaxes, we're back in sort of the other stage. You can see, for example, so just an illustration there of their respective sort of balances, okay? And basically that parasympathetic nervous system, as I said, can be involved in helping the body relax and sort of recuperate from that scary fight or flight response, okay? So think about the issues of respiration, of perspiration, you know, sweaty palms, you know, those types of nervousness. Okay, that's going to be that sympathetic nervous system activation, versus when afterwards you need to make that stuff go away, right? If you're on like a first date with somebody, you want to make sure that you tamp that system down, right? You don't want to be like all embarrassed and nervous and sweaty on your hands, and right? So yeah, you will see sort of those systems are sort of working in balance, and they operate through different neurotransmitters. All right. So, for example, acetylcholine would be one system that might, or one neurotransmitter that might underlie the parasympathetic nervous system versus norepinephrine, the sympathetic. And I'll show you some illustrative reasons or ways in which you can remember that. Maybe if we get to it by the end of the class today. Okay, so that is that there. I'll get to that later. Actually, we'll return to that idea. So. Let's talk a little bit about neuroanatomy for the rest of the class. As we move more into basically the brain from our periphery and then through, we talked about the spinal cord, and now we're sort of rising up. We're going to talk more about the brain. There's a lot of different ways that we can characterize the brain, and indeed that people do characterize it based on different developmental features, based on different morphological features, and certainly different functional features that it seems to exhibit. So we've talked about the spinal cord. There's a couple different parts of the central nervous system as well in the brain. Basically, we'll review the medulla, the pons, the midbrain, the cerebellum, which is a sort of green area here. It's Greek for little brain. And it turns out there's actually more neurons in the cerebellum than there are in the rest of the brain, which is wild. It's where those Purkinje cells can be found that we talked about morphologically last class. And it's involved in a lot of things involved with motion, motor control, balance. And cognition. We don't like to talk about it because we see it in all types of cognitive tasks. We just don't know what to make of it. But it's very much involved, and for the most part, it is a very understudied part of the brain. So if you want to carve out a little niche for you in the field of your own research, go to an area that we don't know much about, such as the cerebellum, relatively that is. Anyways, so that works together with the basically the hindbrain. We'll talk about the diencephalon, basically the thalamus. And the cerebrum, basically the cortical areas. So this is sort of an overview of some of the areas that we're going to touch upon, we're going to look into, and we're going to take sort of a look at how that works. And we're going to look at it within a broader system as well, including some of the cerebral vascular systems that supply the oxygenation and help clean out aspects of the brain, as well as the cerebral spinal fluid as well. So basically, we have ventricles as well with working together as a system of these different areas. And the ventricles are basically fluid-filled caverns. There's a couple of them, they connect, and those fluid-filled caverns are basically filled up with cerebral spinal fluid, CSF, and is also heavily involved in neurogenesis and the birth of new neurons. They migrate from there developmentally. So sort of an overview there of what we're going to be looking at. Okay, we've introduced these ideas of the different hemispheres already, and we'll start at the gross basic level of the brain, and we can start by identifying that we have some very notable features, which makes it really easy to start off with. We can divide the brain into several lobes. First, we divide it in half, in the hemispheres. There's a left hemisphere and a right hemisphere, and they both look pretty much the same, kind of mirror images of each other. From there, right, we can subdivide it, and we most certainly will. So we can identify a couple core features. So one, there's a big line that runs across this way. All right, we call it the longitudinal fissure or the Sylvian fissure. Okay, and that's going to be separating this lobe back here that we call the temporal lobe from the other lobes that we basically identify as the frontal lobe. Anybody know why we call it that?
No.
Yes, I know, guys. Yes, it's in the front. Exactly. So we don't come up with a lot of real creative names for some of these areas. And basically the frontal lobe is delineated from the parietal lobe based on this gigantic line that we basically call the central sulcus. Okay, so this introduces some terms to us as well. Sulcus, right? So the sulca, a sulcus is basically one of these valleys that we see between these folds. So what we can identify then is that we have what we can identify as a cortical sheet. So we have a sheet of paper here, just like you have a sheet of cortex. All right. Now, if you had to get this sheet of paper into your pocket, what would you do? Would you go to the tailor and have them make a bigger pocket?
Most likely not.
Most likely not. You could, right? It costs you a little bit of money, and you'd be walking around with really weird pockets, right? So, what would be an alternative to that?
Just fold it.
Hold it.
Okay, you could just hold it.
No,
Okay.
hold
Fold
it.
it.
Hold
Okay.
it. Sorry. I'm on.
Right, we can fold it. So, right now, when we fold it, we can see that you can fit a whole lot more. Where you can fit the same thing into a whole lot less space, which allows you to fit a whole lot more of that thing into the same space. So we have these folds, right, within the cortex. So cortical sheet of paper, you have the folds. There's mountains and valleys, right? You have mountains and valleys. There's peaks. There's little banks. And the peaks, the mountains, are a single one is a gyrus. Multiple are gyri. And the little valleys, a singular one is a sulcus, and a plural are sulci. Now, one way that I remember that, doesn't mean that it's helpful for you, but maybe it is if you're sad and you're down in the dumps, what are you doing?
Sulking.
You're sulking. Because you're down, you're lower, right? So you don't get, find yourself forgetting and whatever. Actually, funny story, when I was an undergraduate, we had a professor come in for a job talk. And they ended up getting the job, by the way. But he was nervous when he gave a job talk. It was like an hour presentation, and nobody's perfect. We make a few mistakes here and there. And he ended up confusing gyrus and sulcus in his talk. And which is like a really fun, it's just a really simple intro class type of thing. And one of the other professors in the department kind of tried to gently correct him on it and ask did you mean this and this? And he doubled down and he's like, no, it's this other way. It was just one of those facepalm moments. But he got the job. But you don't want to do that. So if you're ever caught, think about sulking down in the dumps, and that's the sulcus. And so anyways, we have those things, and these features help to delineate some of these lobes, and in the back, of course, is what we call the occipital cortex. We'll later find out why people call it the visual cortex, because it obviously seems to be involved in those sorts of activities. And this is about, just to say, this is also a lateral view of the brain. We can look at and see these aspects from a medial point of view as well, and then we can see the cerebellum, basically the spinal cord will be rising up through here, connect through the brain stem, get the pons, and we'll get into some of those other anatomical details. Okay? So we already mentioned these names, so you guys are familiar with them, and we'll spend a lot of time in this semester talking about their associations with respective functions and processing. So for example, occipital area tends to be involved in visual processing, oftentimes we call it visual cortex. Temporal lobe is involved in lots of things, some of which including memory, hearing, language. So a lot of times we call it auditory cortex at areas. The parietal area is kind of oftentimes referred to through functional references, basically association cortex as well. So it's involved in associating a lot of more complex integrations between the other cortical lobes. And the frontal lobe does a ton of stuff too, some of which include higher order cognitive processing, things you might think of as reasoning, decision making, but also you might notice that it's the same areas where we identified Broca's areas before, so language again, speech, but also speech production in terms of the involvement of motor activity. Yes, sir.
Something I was curious about. I was told a while ago, quite some time ago, that the parietal is it seems to light up a lot when it comes to tracking objects. So if it's a baseball coming your way, seeing something move in air or whatever it is that still true for the most part?
Yes. Yes, the parietal cortex seems to be involved in lots of things, which is why I refer to it as like sort of association cortex there, or integrating a lot of the associations with the other sensory features. Now, a point of clarification on the question being that it seems to light up a lot. So, remember we talked about brain myths, they only use 10% of your brain type thing earlier, right? So, no parts of the brain ever light up.
Right.
I've looked. Haven't seen it yet, doesn't mean it's not there, but what we refer to, because we all say light up, is basically referring to neuroimaging activations. And even then, because we do not use only 10% of our brain, usually during tasks, all parts of the brain are lighting up.
Right.
All parts of the brain are usually evident as active. Now, what we refer to when we have those conversations instead are the areas where they're active at a higher threshold. So just a point of clarification when you're with those elevator conversations on the airplane or on the bus ride or whatnot, remind people that we use a lot of our brain a lot.
Do we know how much? Just all of it?
You know, it's sort of becomes sort of an esoteric question, right? Which then is why I have to give these esoteric answers. Because in the easiest example I can use to sort of capture that is it's kind of like having a Ferrari. Okay? So if you have a Ferrari and you're driving it around town, you know, and you're going to pick up a Slurpee at 7-Eleven or whatever, how much of the Ferrari are you using?
All of it.
Except for certain gears, right? Because you're not using like the super high gear. But you're using all the car, right? And if you're speeding around the Autobahn, how much of the Ferrari are you using? Except for some of the low gears. Right? I mean, so how much do we know how much of the brain are you? Well, on what task? When we're sleeping, when we're awake? You know what I mean? Like, we'll learn all these systems that are involved in doing all these other types of tasks. And it's like, yeah, like there might be a time when if you're sitting in the dark, right, pondering your thoughts, maybe the visual cortex is not going to be as active as it would be in another situation. So it's less active, but I mean, it might still be involved in imagining. You know what I mean? Like, so, yeah, it's a really hard question.
It's not like there's areas we don't use or just kind of
like the appendix.
Yeah.
Is there an area that we could do away with?
Yeah.
I mean, not that I'm aware. I mean,
Yeah.
so, well, it turns out, like, yes, there are areas, because we can look at evidence of clinical patient studies of people who lose brain areas, but yet they're not fine, but they manage, right? But again, it comes down to the difficulty of the question, right? So, like, we talked about Gabrielle Giffords, the congresswoman from Arizona, right? She is not fine. She has recovered a lot of things, but there's also struggles and deficits and things like that. So, is that the same thing with vision escape? Is that area of the frontal cortex of the brain, did you need it? Well, it depends on the question. Did you need it to do what? You know, do we need it to get along in everyday life? Probably not. Do we need to get along well? Yeah, kind of. Right? You know, I studied patients who have hippocampal damage. You would never know it. they walk or have jobs, they work, you know, they don't get lost. They're not, like, you look at them and be like, yeah, they don't need that part of the brain. And then I would invite you into my office, and I would try to persuade you with some data that would speak to this possibility that maybe they do need it for one or two really specific tasks in memory. That might be really important types of memory, but yet the other ones don't need it. So it's, you know what I'm saying about
Yeah.
question.
But isn't that more evidence towards neuroplasticity, and not that they don't need specific structures, it's just that the brain will make up for
Well,
it
that's a
and
really good
adapt.
question. So is it evidence for neuroplasticity, or does it speak to the amazing role of neuroplasticity? And I would parse that down a little bit too, because neuroplasticity is the ability of the brain to change and adapt, and perhaps continue to learn to do that function, right, after there's a deficit. But say, for example, the patients that I would try to persuade you with, some data that I've looked at, those abilities are never plastic. They don't come back in those patients. But I said you would never know that they had any problems, because there's compensatory mechanisms, which is different than recovery, right? So it's like, am I being too wonky?
No.
Okay. But they're important delineations and clarifications. Because you guys will be the experts that people will ask these questions to over Thanksgiving dinner when your parents want to know about, oh, well, what about this? And Aunt Sophie had a stroke last year, and is she going to recover all of her functions? And, trust me, you will be out, like, that'll come up, that'll be on you. And, I'm sorry? Yeah. So, anyway, yes, sir.
Just with that, in that realm, kind of, somebody who speaks ASL natively and somebody who learned it, who's learned it, if they have that damage to the Broca's and Wernicke's, will it affect them both? Will they affect those folks differently, or will those that learned it natively, will it be affected similar to somebody who learned language natively?
That's a really good question. I don't know.
Oh.
I'm sure somebody knows. I haven't looked into the role of ASL and deficits to those areas though. We can find out though.
Because wouldn't it be more the motor cortex, moving your hands, and understanding language would be the Wernicke's area?
In theory,
Yeah.
but I mean,
That's why I said native versus non-native.
the world is full of really, really, really good guesses that turned out not to be true. So we need to verify it and check it out. Yeah, I mean, it's very probable and possible. I'm just saying that I'm not going to lie to you and pretend that I know the answer, because I don't. Okay, so we'll talk a lot about these different respective functions that we seem to see evident as described in these different areas and cortical lobes. We mentioned that there's medial views of this as well. Okay, so this is more looking from the inside out of the brain, so this is the midline here. Okay, we can still see these lobes: frontal lobe, parietal lobe, occipital lobe, cerebellum, spinal cord, brainstem, pons, and then there's this little area in the middle called the midbrain. We'll get to that a little bit later too. The diencephalon, where our thalamus is. We'll see the hypothalamus down here. That pituitary gland that Descartes was wrong about. Okay, and we can see down here this inside part of that temporal lobe wrapping around. Okay, so just different ways to conceptualize these. You see this white? What's this white lobe?
Corpus callosum.
You should have corrected me. It's not a lobe at all, right? Corpus callosum. We talked about them as axonal connections between the left and right hemispheres. And when we talk about decision making and things like that, we'll refer to this anterior cingulate cortex. So this is part of the frontal cortex that wraps around in this sort of cingulate, as they call it. So we will look in a lot of this stuff in depth. This is a ventral view of the brain, or looking up from the bottom. Okay, and this is basically the frontal part. This is the posterior part back here. Okay, so this is basically the back and the front. See that spinal cord will be rising back up through here again. Okay, so just sort of orienting to a lot of different areas, because we'll look at things in all these different views in this course of the semester. We want to make sure that we're all on the same track, same page and understanding. And really what we need to do is have the abilities to seamlessly interchange are views of the brain, or we're talking about a process or a structure or a function, without skipping a beat. So we can look at different, views and know that. So here we see the bottom side, basically, of that temporal lobe, the bottom side of the cerebellum, the frontal cortex here. Here we see some more of those cranial nerves that we referred to last class. Okay, you got the olfactory bulbs there. You got the optic chiasm. Those come in there. So just sort of orienting to where we have. We talked about the sylvian fissure and the central sulcus already. And we also can talk about this central sulcus as dividing the frontal and parietal lobes, but we'll introduce those ideas that we'll return to in more depth and specificity when we get into the sensory and motor lectures. Basically, the idea that we have our central sulcus here, and then we have a big gyrus that's very prominent in front of it, and another big gyrus that's very prominent in back of it. The one in front of it, basically the one in the the one in the frontal cortex, seems to be associated with functional activity for controlling our motor movement. This is where we talked a lot about Sherrington doing some motor stimulation control studies and mapping that pre-surgically. And we can see that there's a correspondence as well on this posterior one, or gyrus, that seems to be similarly involved, but in the somatosensory domain. So basically feeling things as opposed to moving things. Make sense? We'll look into that in a lot more depth as we get into more detail. But these sort of anatomical landmarks, like the central sulcus, all right, gives us some other big landmarks by which we can kind of navigate around the brain. That is the motor cortex, the somatosensory cortex. Okay. All right. Okay, we talked about this already. We can take another look at that corpus callosum. So we looked at it in this way beforehand. Remember I asked what this white lobe is of the brain? It's not a lobe at all. It's part of this sort of connection between the left and the right hemispheres. One easier way to conceptualize that is by this drawing. And so what you do is you take this sort of diagonal scoop in theory. So you have an axial view here, looking down on top of the brain. That's the front in there. That's the back here. So you have anterior there and posterior back here. You scoop through, and we can see all these rich white matter projections connecting the left and right hemispheres up to the cell bodies, controlling a lot of that activity in the cortical sheet. Okay, so that reminds us again of this idea of the cortical sheet is this sort of top layer. It's connected to all these other areas by virtue of these white matter connections. And we'll also look at it in a coronal slice, which is evident here. So again, chop my head this way, swing the door open. Look here. I tried to draw this last week on the board. It was really bad. But this is a nicer drawing. Okay? You have the ventricles here. I drew them as little triangles on the board. And we can start to see all this white matter here, and of course the gray matter that wraps around on the outside. Okay? And this just shows where that slice here is taken from. Because it's going to look different based on where you slice it. Because things change shape, just like we saw the slices change the respective views of the balance between, say, white and gray matter in those spinal cord sections. Okay? Okay, so we'll talk a little bit, just introduce some terms of navigation that we will use to guide us through some of the anatomical areas. Okay, so I referred to a lot of these already. If I'm going too quickly, please tell me to slow down. But I'm assuming everyone is able to, we're all on board. Okay, cool. So, we're talking about different planes looking at things. We've talked about axial planes looking down. We just introduced the idea of a coronal plane. Sort of slicing this way, swinging open. Okay, so coronal, you're looking basically at the eyeballs, kind of that direction. And maybe back there and there. And so we can talk about some different directions. Okay, we have coronal, which we've already identified. Sagittal gives us this medial look of the brain, which would be slicing it that way. Okay, so that's slicing me here, and then swinging open the other way. And then, of course, we talk about in the horizontal plane, that axial view that we can get. Okay, So we're going to see pictures in all these different orientations throughout the year. And you can look at the same structure in really different ways. So the corpus callosum is one example of that we've already seen. We can see that in other ways too, in terms of basal ganglia, thalamus. So for example, we see this sort of big sort of oval shaped thing here with this white sort of strip through it, the basal ganglia. Okay, there, and that's the same sort of basal ganglia that we see in this more triangular look here with that same white sort of strip through it. It looks very different because we're looking at it in a very different way, right? But it's just a difference in perspective. And perspective is just the angle that we look at the same reality through. Any questions? Good? Okay. We've done that. Okay, we talked about some of these planes. These are just basically examples of that same thing. So if you remember when I drew that example of the coronal slice before, right? Very simple conceptualization. I use these little triangles to represent the ventricles, right? And I added the cortical folds. All right, we can see that these ventricles are going to look this way in the coronal slice, but here they look a whole lot different in our horizontal slice. Same things. And then, because indeed, when we look in the other direction, medially is the easiest way to look at those. So, right, if this is our brain looking sort of or lateral or in the sort of sylvian plane, we can see that the ventricles themselves actually follow a C shape, sort of like that, which is why it's going to change based on which angle and orientation that you look at it. It can look very different. And that shows some of that same triangle. So this is this. Shows you how bad of an artist I am. Okay. So a couple terms that will be used through the course. We talked about anterior and posterior last class. Post means the back of or the end, like the post-game show after a basketball or football game. Anterior means in front, because sometimes you'll see that interchange with rostral and caudal, respectively. Okay. Inferior means sort of lower. Superior means a little higher. So you might talk about that in the context of, let's say, this might be superior here, and a little bit lower down here might be the inferior parietal cortex. All right, it's just a way of telling people when you write a report or you're reporting back to another person or scientist what you found when you looked somewhere. Where did you look? And where did you find it? Well, it was in the parietal cortex. Great. Big area. Tell me where I should narrow my search down. The top end of it, the bottom end of it, and they like to spruce it up by calling it superior, inferior type of thing. So you'll see that ipsilateral and contralateral, respectively, refer to either the same or the opposite side. So we talked about this idea earlier in the class that the right side of the brain was involved in, let's say, controlling the motor activity on the left side of the body. Okay, that would be contralateral control. But there's also ipsilateral control too, which means the same side. Which means there is input from the left side, the left hemisphere, that is the brain, controlling aspects of the left side of the body too. And of course they work together. And we've already alluded to the idea of medial and lateral. Medial, of course, meaning more towards the midline. Lateral meaning more towards the outside. Okay, so you'll talk about the lateral parietal cortex versus the medial frontal cortex. Okay, and it'll just be expected that we know where that is, because we can walk through that navigation in our mind in terms of where it's referring to. Okay, so here's some other ways to visualize that if you need to. Notice a couple things that change. Where does it change? Kinda, yeah. So what? Yeah. So we'll remember it'll be a little bit more towards there. But what changes here is our axis, our plane. So you have a plane that goes this way, but also a plane that goes this way. And so you'll see some anatomical slides, let's say in the spinal cord versus the brain slices, that might look like they're referring to different things when you're talking about anterior, posterior, dorsal, and ventral, things like that. And the reason is because a lot of some of those terms, or at least come from the rich history of anatomy in animal studies. Okay, so let's say we were one of the few species that stands up straight. So the spinal cord used to just follow straight out from here. And we never had this rotation. So you can look at like a rodent's brain, which is a very common neuroscience species. Here we have dorsal, which should mean dorsal. But instead, dorsal means like towards the back. That's why I was saying here to show dorsal versus here when we were talking about the spinal cord, right? But we can see that it makes sense when we just exhale, take a breath, think about it. Okay, that makes a lot of sense. We can move on from there, right? So know that sometimes we'll have to change that orientation as well, based on what we're talking about. Does that make sense? Okay. We got that down. Let's try some examples. So this is a frontal view of the brain. That's the anterior part. Let's see here. What view of the brain is this?
Ventral.
Yeah, ventral. Okay. What side of the brain is her hands on?
Posterior.
Yeah, on the posterior side, not the anterior side. So anterior would be up here, and Lindsay was right, it's definitely a ventral view of the brain. Okay, so what view of the brain is this? Yeah, it's sagittal. Exactly. So you have two definitions, two references here. You have both mid and sagittal. Why? Because you can equally have a sagittal view of the brain that's all the way out on the side here. How do you know it's mid?
Straight down the two hemispheres.
But how do you know that that's what this? How do you know that's where they took this slice from?
You can see the internal structure.
Well, you can see the internal structures of any slice.
Looks like the corpus callosum.
Okay, so corpus callosum is a good identifier. Think about really big landmarks.
You're able to see the frontal, the opposite to the one you're able to see.
Well, you can see that on the lateral side too.
Yeah. Fissures?
How about this, like the spinal cord coming, or like the hindbrain? Like this is in the middle,
Yes.
right? It's kind of like, so like my neck here is like more towards the middle, but there's parts of my brain that would like hang out more laterally beyond my neck. So if you can see this thing in the middle, you know you're much more medially as a starting point. So yeah, all good points by the way. All right, so we can also talk about proximal and distal. Okay, distal, distant, very far away. Proximal, much more closer. Okay, we'll talk about that in some motor control lectures. We already talked about afferent. We talked about efferent. Okay. Okay, we've already talked about gray matter and white matter. We can just show some examples of that a little bit more in depth. So we have, to take a microscopic view of gray matter, you can see a lot of cell bodies. Okay. We also have white matter that we can see, right? Myelinated axons. If we zoom in on white matter there. So some examples there. Let's see here. So we're not going to talk a lot about development in this class, but would be remiss if we didn't mention it a little bit. So some of these areas we can see develop in different stages in terms of timing. So basically we can categorize sections of the brain, and their color are really helpful here. Okay, so basically you have the telencephalon, the diencephalon, the forebrain, midbrain, and hindbrain. So essentially here in development from embryo, you basically just have a tube. That tube is going to start to grow, as we all have. We all used to look like this at some point or another. And as it grows, right, just like our paper needed to fit into our pocket, in this case, our cortical or our neural sheet needs to fit into the cranium. And our pocket doesn't have a whole lot of room, and neither does our cranium. So the skull doesn't like to grow very quick. So we need to pack brain stuff in there. And so it starts to fold. As you get more and more days, you start getting this folded, and now it starts to come back up over it. Okay? And you can see the expansion of initially, so you have the forebrain, and then you get basically it develops into this telencephalon, sort of wraps over it, which ends up becoming that cerebral hemisphere. And we'll look in the more developed version of this delineation, basically. So we have our cerebellum, there's the pons underneath it, the medulla, then it comes the spinal cord, and of course our cerebral area. So sort of just the developmental trajectory there. And now we can start to categorize that in those same terms. So here's sort of how they started off, right? Sort of like Drake down at the bottom, right? Or perhaps this is maybe all that Drake might have. If you've ever seen him dance, you might consider that he might not have more than neural tube up there. But it's a conversation for another time. And we can see those same areas categorized, I think, both more helpfully but also more confusingly in this graph and table. You're not going to have to know all this stuff and have to reproduce it in this type of way, but it's here to help sort of tie those lines together. So the colors, I think, are really helpful too because they tie together those same things on a much more anatomically illustrative slide than those neural tubes and just those sort of cartoonish depictions earlier. So we basically have our spinal cord coming in, the pons, medulla, midbrain. So we have the hindbrain here. We'll talk about the structures that compose the hindbrain. We'll talk about those in the midbrain, and then the forebrain that we're going to include things like the diencephalon, the concephalon, things like that. We can see how they fit within there, and then we subdivide into these other structures. Okay. So lots of stuff, lots of colors, all that kind of good things. So mention a few things about the cortex, and we can sort of take a little microscopic slice from there, and we can zoom in on it. This is the top of the cortex. This might be the bottom part before it becomes that myelinated white matter. We can see a couple features. One, that a lot of the cortex is organized in such a way with pyramidal cells. We talked about those pyramidal cell morphologies last class. There's that pyramid sort of in there, that little triangular shape. Our dendrites, our axons projecting up. And we can see that we can categorize, much like Brodmann did, right, the morphology and histology of these cells, right, different sections of the cortex. And there's sort of a theme that seems to resonate in different types of topics of conversational aspects of what seems to be evidence in sort of this principle of cortical organization. And that is that the brain, and indeed the cortex in this case, seems to be like ogres. Because ogres are like what?
Onions.
Onions. Why are they like onions?
They have many layers.
That's it, they have layers, donkey. Okay, so, I'm sorry? And they just smell bad.
It's the formaldehyde.
It is the formaldehyde. So what we can see is there's really, really consistent labeling that people refer to as these different cortical layers. All right. So the top layers, you basically start 1, 2, 3. Then you actually subdivide the layers. There's 3A, there's 3B, then there's 4, 5, 6, and then of course we get the white matter, or their myelinated axons are projecting out there. Now we can see that they're actually, so these are cell bodies. Okay. They're stained basically through Nissl stains, which we'll get to in a little bit, which basically identifies cell bodies, but none of the projections and structures. Here we can use a different type of stain, basically like a Golgi stain, which stains the whole cell, not just the body, but its actual projections, and we can see these layers and the projections among them and across them. Okay? Now the Golgi stain is useful for identifying the whole neuron, but for some reason it only stains 5% of neurons. It's really helpful. Why?
You're able to differentiate. You're able to see one in its entirety instead of all of them tangled.
What would it look like if it stained the goal of 100% of neurons?
You don't see anything.
It'd be black.
Yeah.
Right?
Do you do it that way on purpose?
No.
Or is that just how it happens?
No, Golgi would love to claim credit for that, I'm sure. But my understanding of it is that nobody knows why it only stains 5%. It just does.
Is it like the same 5% like you?
I don't know that one. I just know that it's 5% and that nobody really knows exactly why it only stains a certain percentage. It's kind of like Joe Dirt said about Positrack on a Plymouth. Nobody knows why it works. It just does. Okay. Sorry, I'll throw some movie references in there and try to keep it.
Is a Golgi stain something that's accessible to folks?
Yeah. It's been around for 100.
No, I mean, like, I could go out and buy it technically, if I had something.
Maybe not at Michael's craft store.
Right.
But, I mean, it's more accessible than doing like drug studies where you need FDA approval to, hold LSD in your lab or something like that.
Right. Or cocaine in the basement.
So, yes, which is actually probably a lot more accessible. But it's somewhere in between Michael's and the FDA and the DBA. Yeah. Yes. That's it.
So although the top part is the gray matter, there's still,
So this is all gray matter for basically here.
yeah, but there's still cell bodies up there that have myelinated sheets, or are they
No,
not?
well, myelinated sheets do not surround cell bodies.
Well, the axons that come out of them. It's not like the axons just come down, they're only in the bottom part, right? So the ones in the top are just not myelinated?
Well, yeah,
Or-
so if they're making sort of short-term connections here, then yeah, they would not be myelinated. At least not in any meaningful proportion. Again, like I try to stray away from like statements about absolutism. So it's not to say like you can't come running down the hall one day to me like, look, we found this one cell that was myelinated up here out of like this 7 trillion in the rest of the. Alright, great. But yeah, as a rule of thumb, yeah, I mean, not really. Okay, so we got some of that. We got that. Let's talk a little bit about the forebrain. Okay, so, and this is why we started off, and we introduced these ideas for some of the terms that we're going to use. So we're using the forebrain here. Okay, it's part of basically that basic larger part. It includes the telencephalon and the diencephalon. Okay, so it's basically, when we're talking about the forebrain, we got the cortex, the basal ganglia, the hypothalamus, the thalamus, all those things. Okay, so, all right. So some of the main structures that we're going to introduce, okay, are illustrated here. This is a nice sort of three-dimensional attempt at representing it. Okay, and remember that these are bilateral structures. So it's very difficult to show things bilaterally unless you're looking at a coronal view, and that tends to obscure some of the organization of how things are shown to you. So this looks like it's one-sided, but know that it's two. So you have the basal ganglia, which, remember the word ganglia means collection of nuclei and things like that. So it's actually a collection of cells, and there's different parts of it. You basically have this big chunk in the middle, and then you have this wraparound tail. Okay, so we can subdivide it into further components. Okay, you have the putamen and the globus pallidus. Okay, and each component of the basal ganglia. We'll get into that in a little bit more detail because it turns out it gets a lot more complicated than that. But for beginning conversations, we can just orient to some of the other structures that we have here. Okay, within the basal ganglia network, there's a little green area. It's actually a lot easier to see on a coronal slice, but we can see it here. It's basically the subthalamic nucleus, or the STN, and beneath that is the substantia nigra. Okay, so that's going to be this area down here. When you look at a coronal slice, we'll see some of the ways that they network together. That might be a little bit later in the semester. We can also look at the amygdala here, right, sort of at the anterior medial side of the end of the tail of the caudate. Okay. Let's see here. So this is one way that we can look at that coronal slice of it as well. Okay, so we basically have our ventricles here. Okay, we know that we have our landmark of our corpus callosum connecting. Okay, and the basal ganglia is basically being shown between the caudate and the putamen. Okay, so that tail of the caudate, remember, is wrapping around. The putamen is going to be down here, and it's composed of basically subcomponents of basically the globus pallidus internus and the globus pallidus externus, which is divided by some white matter across through. Okay. So we will look into that in more specificity as we move along. We can look in some other aspects of an area, or a system called basically the limbic system, which is basically refers to something that needs around the rim. We'll talk about that in more depth too, but this is sort of just introductory today. In the limbic system, some of the primary components that people typically refer to include the hippocampus, the amygdala, things of that nature, although the actual set of structures that the limbic system refers to has changed over time, which is a royal pain. So limbic system isn't the most common way that people refer to it in the modern conversations, but it's a really common way that people refer to it nevertheless. It's just not like, it's more like of a textbook reference than it is like journal article papers. Does that make sense? Okay. So generic as opposed to specific. And we'll talk about some of those others. Other key places, the cingulate gyrus, we talked about that before, we looked at it on that medial slice. Okay, that sort of about where the anterior cingulate was, sort of wrapped around, and that, we'll look at that in a minute too. And it also involves access to the olfactory bulb, part of those cranial nerve systems that we talked about yesterday. Okay, so here we can look at it in kind of a way. Okay, so we have this sort of hippocampus wrapping around the green. Okay, in front of that is this blue area called the amygdala that we introduced earlier. That's sort of an almond-sized shaped structure. Okay, the anterior medial part of the hippocampus as it wraps around. Okay, we also have essentially that cingulate cortex here that we talked about. Okay, that can be subdivided into the anterior part, the middle cingulate, and the posterior cingulate. Okay, our thalamus is in the middle here. Remember, that's a bilateral structure. Okay, so you'll see one of those on each side. The thalamus is basically serving as a relay station for a lot of our sensory projections and processing. So a lot of the information, be it visual, auditory, sensory, before they go to the cortical areas that are involved in processing them respectively, they basically go through the thalamus first, which is kind of like a changeover station for like a train. So it's where you transfer trains, information comes in, goes and projects it out to the right spot. So the thalamus is very, very important for that, and hence it's very important for just about everything. This just illustrates some of the olfactory bulbs, the projections into the limbic system by virtue there, and the mammillary bodies are basically part of what's wrapping around the fornix. Basically, the extension of what ends up becoming the hippocampus, these fornix and the mammillary bodies over there. Okay. So, some orientation to terms and things like that. We talked about thalamus. Oh, the hypothalamus, I should mention too. It's this sort of yellow area down there. Can you guys see where the pointer is? Okay. So we have that yellow area. The hypothalamus is involved with a lot of basic evolutionary functions. We'll talk about that in the context of motivation later on in the class and regulatory processes. It also controls the pituitary gland that we introduced from Descartes' inferences. The hypothalamus is, yes ma'am?
Is that not the pineal gland? That's the
I
pituitary?
confused the P's.
Oh.
You're absolutely right. Yes, the pituitary, though, is involved. Instead, it was not the seat of the soul that Descartes thought. Good catch, Lana. Pituitary is involved in a lot of hormone regulations and things like that. So, the hypothalamus is famously involved with the basic sort of endocrine functions and things regulating what people have tended to refer to as the 4 F's. Mm-hmm. So, fight or flight,
food and sex,
feeding, and reproduction. The 4 F's. Okay, so this is a different angle of view to look at some of these limbic system structures. We talked about the basal ganglia, for instance, before. We can still see that serous neurons coming through here, globus pallidus, internus, externus, things of that nature. So these are just illustrative. Let's move down a little bit into basically the midbrain, okay? And the midbrain is a very small area of the brain, okay? It's basically this sort of little area. So we've got this midbrain there, okay? It includes little things like superior corpus and the inferior corpus, which are very difficult to see here. But if we look at it in another way, right? Let's say, all right, you have your brain here. You have the spine coming up. Arms, midbrain. Good. All right. When you look sort of internally, that stuff now. There's basically a bump here and a bump there. That's the superior colliculus, that's the inferior colliculus. Inferior is involved in auditory function, and the superior is involved in visual processing. We'll talk about that as we move along too. We talked about the substantia nigra as being part of the basal ganglia system. Okay. That's going to basically be, again, in this midbrain area. And some other areas, the reticular formation, periaqueductal gray, tiny little nuclei, again, sort of within there, and then you have the reticular formation where basically nuclei going down there. Let's see if we can cover some other things. We've already introduced the idea of the cerebellum, which of course is this back area there. It's a very, very elegant system that we understand very well. The anatomy is beautiful, I happen to think. It's a subjective judgment of beauty, right? But I think Purkinje cells are really cool looking. And when you actually break down the anatomy of the cerebellum, it's really quite complex and very, very cool. Has different lobes in its own right, those types of things. So we have the cerebellum, involved in body memory in its own regards. And of course the pons and the medulla. So the pons is going to be this big bulging sort of area there, and the medulla is sort of that connection, the area between the pons and the spinal cord within this sort of hindbrain area of the brainstem. So that's one your medulla there. The medulla is really involved in basic life-sustaining functions. Its nuclei control a lot of things like respiration and heart rate and things like that. And when you have issues like pressure on the brain or water on the brain or other types of tumors that press on stuff, the brain can't expand because our cranium won't allow it to, right? So stuff has to compress down, and these are areas where when they're suffering a lot of that pressure because there's growth of tumors or other things like that, pressure from water or cerebrospinal fluid that won't drain, these are times when it can be fatal because it might compress and damage some of these basic, very primitive areas of the brain that control life-sustaining functions like respiration, heart rate, heart beating, and obviously when those things are damaged and screwed up, that's no bueno. So we have that there. Okay, we've talked about these other views. So I'll post all these slides for you guys anyways, just so you know, a lot of the slides that I have on here are just for viewing, just so you can look at it in different ways and angles. So we talked a lot about that basic orientation sort of tour then through some of the main primary structures. What I want to just touch upon and visit before we finish up are some of the associated areas and structures and different anatomical components basically in and around the brain that provide sort of the larger system that the brain operates in. So basically we'll talk about the meninges, which are the wrappings, so you can think of, they look like this. So we tend to look at brains like we just did, right? But it's actually, those are isolated brains. A lot of times they've had the cerebral vascular aspects pulled off, right? So all those cartoons you just looked at, they didn't show the blood vessels. They're there, you can see them, the blood vessels in there, okay? This would be the spinal cord, and they're actually all wrapped up within these different layers of protective, not skin so much, but they're protective membranes, okay? So we'll get to that, and then I'll allude to these other ideas of blood-brain barrier. So we have about 10 minutes, and we'll see if we can knock all that out. So, I should say too that these pictures were taken at when we were actually doing these anatomical dissections at a couple different opportunities that are great options for graduate students to apply to. So one of them where some of the other people who were holding up brains, we did that little quiz of what angle is this being shown at. That was from the Woods Hole Marine Biological Laboratory. It's a summer program in neuroscience, ethics, and survival that's designed for graduate students. So basically 2 months on the beach in Cape Cod, all expenses paid, free food, room, board, yeah, pretty awesome.
He had me at food.
They had me at free.
Yeah.
And so it's a really great program. If any of you guys were here last year and knew Dr. Iniguez, him and I were actually both students there together. That's where I met him. Really great guy. If any of you guys might have known him as an undergrad. But it's designed for graduate students. To go there, you learn a lot about neuroscience, neuroanatomy, a bunch of classes, but also professional ethics, professional survival skills, how to do grant writing, how to put together posters, how to successfully navigate conferences and meetings and presentations and job markets, and build a mentoring network that is really helpful to career success. And
What's it called?
I'm sorry.
it's abbreviated easily as SPINES, but it stands for Summer Program in Neuroscience, Ethics, and Survival. It's at a place called Woods Hole Marine Biological Laboratories, which is where they actually isolated the axons of the giant squid in order to discover the action potential. It's a very, very famous and prestigious institution because it has a lot of Nobel Prize lineage there. That's where Hodgkin and Huxley actually did some of that work. And it's a marine biological laboratory because it's a giant squid axon and access to the ocean through some of those mechanisms.
How do you say that?
You apply. Yeah. So I'll see if I can put a link or something like that up. Usually when calls go out for that a little bit later in the fall, put that application up there. And anyways, you got to do some brain dissections. And another one, if you're interested more on the cognitive side, is just the Summer Institute for Cognitive Neuroscience. So that was at University of California, Santa Barbara. So it's sort of like Tupac and Biggie, of East Coast and West Coast. And you know, this is the West Coast side. Then you see Santa Barbara. It's going to be more cognitive and less so about professional ethics and professional survival skills, but definitely the same type of neuroanatomy and high-level science and presentations and things like that.
And that's summer too.
It is summer as well. Yeah, they're great. They're great gigs. So they're anyway. So I just wanted to put that in there so you guys can keep that on the radar. And that's where we got these pictures from. So this is actually us chopping these brains apart and stuff like that. So it's
Are
pretty
they
cool.
sheep brains?
No, that's it. That was a human being.
Really?
Yeah. Yeah, so that's what this is too. This is just a ventral upside down view, the cerebellum. Yeah, it's a big old sheep.
That's big.
It's a really smart sheep. No. Okay, so let's take a look at some of these other features while we still have a couple minutes. So we've talked a lot about the brain, but of course, the brain is operating with the larger system. So it's encased by the skull. That's what this bone is there. And between the bone and the brain is a handful of layers that's broadly called the meninges. And the meninges themselves can be broken into several different layers. Basically, the pia, the arachnoid matter, and the dura matter. So dura, Latin for tough mother. All right, so it's the tough outer casing because God help us if you were in battle back then or now and your skull was opened up. Wouldn't you like a little secondary level of protection so all the dirt from the ground doesn't get kicked into here? Yeah, of course, right? It's very important that nature has built in a lot of protective mechanisms here. Okay, so of course, first is structurally, the skin and the skull, but now we're actually got these layers of protection, the meninges. So the dura, the pia, and the arachnoid space between. And of course, embedded within them are the blood vessels, okay? The blood vessels are going to be obviously delivering blood and returning blood back to the system after being deoxygenated, and as well as cleaning out some, bringing nutrients and cleaning out certain aspects of detritus. Now, the blood we'll talk about in a little bit is very good at delivering oxygen and certain nutrients, but there's something called the blood-brain barrier, which means that certain chemicals cannot pass through because it's a physical barrier. Okay? Now, it's a mesh, so you can think of it as mesh. Certain molecules can pass through if they're small enough, but larger molecules can't. It's a big problem for drug delivery. If you're interested in pharmaceuticals, pharmacokinetics, a lot of drugs that we synthesize cannot pass through because they're too big of molecules. Which is why it's very difficult to treat certain conditions and why we have to come up with these creative workarounds, such as delivering the smaller molecule that is the precursor that allows the bigger molecules to be made inside the brain, which the smaller molecule that's the first precursor gets able to cross the blood-brain barrier, gets in, and so it makes things hard to do in that regard. So, like I said, dura matter, tough outer layer, pia matter is much more delicate. Okay. The arachnoid sort of space and membranes in between there. So that's a cartoon version. Well, they're both cartoon versions. But you can see those respective relationships there. Okay, this is a real version of those meninges. You can see some of the blood vessels contained within them as well. Okay, so we've kind of worked peeling this off. So here we can see that meninges is taken off, and we see that traditional look of the brain that we're used to seeing and expecting. But in reality, it looks more like this. Right? We talked about that difference between the sort of our conceptualization of things and what reality is last class, and this is kind of reflecting that as well. So here's that it's wrapped up, wrapped off. You can see that there. Here we see some other sort of examples of pathologies of the meninges, such as tumors and other things like that. Meningitis, inflammation of the meninges. Right? Because within the central nervous system of the brain, we can understand why it's such a serious issue to be concerned about when you have something like viral meningitis. Yeah. So you might see our normal little slice. We have blood vessels running through the cortex, the skull, and our meninges sort of connecting there. When they're inflamed, we can see the problems thereof, the constriction of some of those blood vessels, all those sort of problematic pathologies. Okay.
Does that just randomly happen, or is it genetic?
Well, a virus is going to be something from the environment.
Okay.
Yeah. Now, the complications happen in that, there can be genetic variation sometimes in how we respond to viruses. But that's a whole other conversation. But yeah, if it's viral, it's going to be generally from an external virus. And then of course meningiomas. Jomas or omas are typically referring to some type of tumor in one way or another. And we can see one example of that here clinically. If you pull back these meninges, here's one big old tumor there that was otherwise depressing and resting in there. Now you can imagine deficits that are very understandable then that you would have when you have a tumor like that depressing stuff like that in the brain. So the way that neurologists work a lot of times is they have to work backwards based on symptoms and see, what are you having problems with? Is there a spot in the field over here that you can't see? When we trace that back, maybe that can tell us about which area of the visual cortex there might be a tumor that's compressing and pushing pressure on that those neurons aren't working really optimally. Make sense? And of course, you did the same logic, but for frontal cortex and things like that. Okay, we got 2 more minutes. Let's see what we can knock out here. We mentioned the ventricles. I did promise we'd return. So here's a lateral view of them, that C-shaped structure. Okay, we have basically there's sort of these different horns of it, the frontal, occipital, and it wraps around, sort of like our cortex does, this temporal side. Okay, so that's our sort of main ventricle there. We have the third ventricle. Okay, this more central version, and then it goes through the cerebral aqueduct to the fourth ventricle. Okay, and then it goes down to the spinal cord. Now within these ventricles are where they make the CSF, the cerebrospinal fluid. Okay. And the lining of the ventricles are called the choroid plexus, which is what actually makes the cerebrospinal fluid. Here's different ways to look at it. Clinical deficits or conditions of disruption of this CSF system of the ventricles include hydrocephalus, basically water in the brain, or overproduction of too much CSF, or blockage of the drainage, in which case clinically your doctor might use a shunt to basically drain some of that fluid. So we can see illustrations of those different ventricles there. Illustrations of clinical hydrocephaly. Okay, the growth of some of that because the skull is still somewhat flexible during the younger fetal development, postnatally. We can see they can actually grow that here. You can see normal ventricles versus the larger ones. Okay, so, and briefly we have the blood vessels. I will leave you to visit them here on your own, but we talked about them a little bit with the circle of Willis on the first day of class, and we know that we can categorize them by the same anterior, posterior, middle artery classification systems that we talked about orientation-wise, and you can read a little bit about how their rupturing and blockages can result in stroke and things like that. So that kind of wraps me up for today. We've already talked about the blood-brain barrier, and
Is that you on Kilimanjaro?
that is me on Kilimanjaro because I had a problem with the blood-brain barrier because at higher altitude, that mesh is held together down here at atmospheric pressure. The atmosphere puts pressure to hold it, but when you get really high up, the atmospheric pressure gets less and less, so those holes in the mesh open up and more molecules pass through. And so when I was up here in Kilimanjaro, I had that happen, and all those molecules pass through. I had cerebral edema and pulmonary edema because it happens in both areas, and you end up, even at the top, getting your tail carried down. It was basically terminal stages, so it's very dangerous. And so that's sort of the last clinical aspect of each of these little conditions that I referred to between the problems of the meninges, problems of the vascular system, problems of blood-brain barrier, problems of the ventricles. So that's all I got for y'all today. Is that enough?
Yeah. Okay, that was good.
Does it just fix itself as you go back down to normal altitude, or do you have
Yes and no.
What day, right?
It fixes it a little bit. Like the pressure will fix, but you've got to clear out all that excess fluid now that doesn't have anywhere to go.
That doesn't happen to everyone, right?
Not everyone, no. Just
You're just lucky.
I'm sorry.
Why are they okay with people carrying these out?
Because they've acclimatized. They're locals.
They're not as lucky.
Yeah, I was very dumb. I went there from carrying on Jersey Shore at 0 sea level,
Oh,
and I went to 20,000 feet bucket.
What?
So-
Aren't you supposed to camp like halfway up for a week? Or
Yeah.
somewhere near the top?
You are. Yeah, you can just put them there. Thanks. These are just, I don't need to give them to you, because I was going to give
I want to do Kilimanjaro someday. Yeah. No little.
Acclimatized.
Yeah.
Thank you.
Should we just take it anyway?
No, I mean, I'll post it online. You can take it if you want.
I mean, I have this phone.
Bless you, bless you.
Excuse me.
Hey,
You gotta put-
I got your email, but you didn't send me the instructions. I'll just
Okay.
get the instructions.
I did in a separate email. What
Yeah.
email are you looking at? 'Cause I'm sending it to the Coyote.
Yeah, I'm looking at the Coyote. What happened was I combined it, right? So I get all my stuff. So I get everything forwarded to my phone, so I'm always in constant contact with it all. Some things didn't translate over for the last couple of weeks, unfortunately.
Okay,
So-
did you get the access to the drive?
Yeah, I did. I
Okay.
was able to fix
I'll
that.
read you the instructions then.
Yeah, so that was the only thing. I was looking at it yesterday because I was trying to see if we had it up there. And I was trying to use one of those examples.
Sorry, I'll get out of your way.
I said, I kind of need that master sheet.
Yeah.
Just to give myself a little bit.
Yeah.
Give you an FYI, I'm okay with it, but some people do get this.
So if you want to get it all
So
done.
the way it works is, it's unclear, but like the class, the next class starts at 12.
Yeah.
Some professors
Yeah, no.
do regularly
Yeah,
at
send it to me.
10
I'll
and set up
Yeah,
the
much like
next
Keith
class,
and
and
Penny,
expect that they should be
get a
able to
little
do
sleep.
some. And I don't know, I feel like we should give each other some latitude. But
Yeah.
I'm more liberal about that.
Yeah.
But I've seen professors have discourse about this before. So,
I will,
yeah, I'm not complaining, that's what I'm trying to tell you.
okay.
But